So thank you very much for having me today, and thanks for coming to this talk. As I suspect a lot of you probably haven't heard of Owlstone Medical, I'm first just going to give you a bit of insight into who we are and what we're doing. So first, a quick fact. As we're actually exhaling hundreds of small metabolites from our body that have high volatility. These can be classed as volatile organic compounds, or VOCs for short. Now, these molecules are absolutely tiny, and they're in the breath, and they're not just reflective of what's going on metabolically in the lungs, but they're also reflective of what's happening across the whole body. So many of these molecules come from the bloodstream, they're very small, they easily diffuse between tissues. So breath is therefore a potential source of non-invasive metabolic biomarkers. Now, Owlstone Medical is a company of, Owlstone Medical is a company of about 200 people, mostly based in Cambridge, UK. And we are basically developing and deploying breath biomarkers and breath tests for ourselves and for partners. So we provide tools and products to help you to collect breath samples if you want to do the analysis yourself. But we also do targeted and untargeted analysis in services for people who want to understand if there might be breath biomarkers of relevance for the diseases of interest to them. So today, I'm mostly going to talk to you about some of the work that Owlstone's been doing recently to make reliable analysis of breath metabolites a bit more possible. So basically supporting the longer-term development of volatile metabolite biomarkers. I thought first it might be useful to give you a few of the challenges that have historically existed in this field, because it's not been an easy area to work in. So on the collection side, you have to think about how to standardize collection of a breath sample to make sure that a breath sample from one person is directly comparable with the breath sample from someone else. Now, as everyone breathes differently, that's actually quite a large challenge. You also have to think about how to minimize confounders. So VOCs are in the air all around us, and they can very easily contaminate samples. And finally, there's very diverse ranges of VOC chemistries and a really wide range of concentrations in the breath sample itself. On the analytical side, you've got some of those challenges again. So you've got the fact that VOCs are also going to be environmental air around us. You've also got the fact that you're working with a very diverse range of concentrations and chemistries. But you also have the fact that actually, when you're looking in breath and you're doing GCMS, which most people do in breath, the libraries which you use to actually identify the molecules at the end aren't all that great. The NIST library is sadly not all that super for breath yet. So what we're seeing is there's actually quite a lot of potentially quite inaccurate identification of molecules and tentative identifications out there. There's also, of course, a complete lack of reference ranges for compounds. So those are some of the big challenges in the breath field, and that's what Owlstone has been trying to help develop solutions for. So the first thing needed, of course, is on the collection side, a method to be able to collect VOCs from breath, avoiding those collection challenges. So Owlstone have engineered a system called the Breath Biopsy Collection Station, which you can see in the center there, which basically allows you to collect VOCs on two sorbent tubes, those metal tubes up there. And what we do is we take a breath sample over a prolonged period of time, and that concentrates the VOCs up, as the VOCs are basically stuck to the sorbent and the rest of the air is passed through the tubes. So by taking multiple breaths, typically over a period of about 13 to 15 minutes, and we can actually obtain a sample of VOCs that are both enriched for very high concentration and low concentration molecules, so increasing the breadth of molecules that can be analyzed, but also, of course, increasing the sensitivity. The system is built also to avoid introducing noise, both through the actual materials that we're using to actually collect the breath samples, avoiding materials that both leach and absorb VOCs. But also there's a unit called the Casper, which provides basically patients when they're breathing in some air that's been cleaned up environmental VOCs. This also comes with a computerized system control the receiver, which allows us basically to collect specific fractions of the breath. Now, we know the lower fraction of breath contains more signal-rich, less noisy fraction. So the system allows us to concentrate on that lower breath fraction and collect that specifically, while monitoring exactly how much volume has been collected so that we can be confident of a sample that's comparable to another sample. And you can see this in this bar chart at the top here. Our system actually allows you to collect two samples at the same time that could be compared against each other. And you can see that actually the variability between those two samples is very, very low. So we have succeeded, I think, in creating a collection system that allows you to collect a very good, reproducible breath sample. To address some of the analytical challenges, we started a project a couple of years ago to really start understanding what VOCs actually exist in breath samples, and maybe aren't just from the environment around us. We also want to know what levels these molecules are at. So our aim here is basically to develop methods that can reliably probe a wide range of biologically relevant molecules, accurately identify those molecules, and quantify their levels, ultimately enabling the easier development of breath biomarkers. Now, we started this project off by taking breath samples from around 100 healthy volunteers. There are a range of ages, genders, ethnicities, and we also collected blank samples. And these were collected with our breath collection system, which I just outlined on the previous page. So the samples and blanks are then analyzed by thermal desorption GCMS. We used GC Orbitrap systems from Thermo because the high resolution and accurate mass basically makes this work a lot easier, as breath isn't a simple matrix, it's got a lot of compounds in it, and we want to be able to confidently identify the compounds. We then deconvolute the data, perform feature extraction and normalization to give us the features representing VOCs, and then before doing any identification of the features, we actually first work out which of these are on breath at significant levels versus in the blank samples. Now, those molecules, on-breath ones, are the ones that are likely to be coming from biology versus in the environment. And then finally, for VOCs that we do think are on breath, we identify the features. Now, we run quite a lot of standards in our GCMS. If the molecules in breath are one of those standards, we can see those and get a very high confidence identity. We'd call it a tier 1 identity. There's also lots of molecules we've run in our library, and so those can be tier 2 identities if they're in that library. And then finally, if they're not in tier 1 or tier 2 and they're not in the libraries or the standards we're using, we can compare to NIST libraries and get an identity there with a tentative nature. Now, of course, we don't want to leave it with tentative. So from the healthy population study that I just outlined, we identified 328 on-breath VOCs, and this is using 3 different statistical methods to compare breath samples to blank samples. And many of these 328 couldn't be given tier 1 or tier 2 IDs, as they weren't standards or weren't in our libraries. And so we've basically, over the last year or so, been going through and trying to identify what these are so that we can add these to our library and then identify and quantify them in future studies. So starting from the VOCs found in a very high proportion of the healthy population, which is the low-hanging fruit, basically, we then worked to identify what these compounds were. So using the NIST matches, guided which potential molecules these might be, we then brought in a lot of standards, and then ran a lot of standards on our GCMS systems, and then manually compared to the original feature to see if we could confirm an identity. If we could confirm an identity, that was then added to our VOC atlas, which is the atlas of on-breath compounds, so that we can look at this reliably and identify it and quantify it in future studies. And so far, we've added over 150 compounds to this breath atlas. These are confirmed on-breath molecules. And of course, we've been now thinking about the biological origins of these compounds since, and we see many of these reflected, when biological processes reflected in these kinds of VOCs. So we see, for example, metabolites that are related to fatty acid oxidation, related to amino acid metabolism, some compounds that have been prior linked to inflammation and things like that, and actually lots and lots and lots of compounds that are related to the microbiome. So we see a lot of short-chain fatty acids, trimethylamine, indole, and things like that. So to summarize, basically what Owlstone do as a company is we have developed breath to be able to translate it to something that can be useful clinically in the future. So we have products and tools that can help to collect very reliable, reproducible breath samples, and services which can be used to profile metabolites and help you to understand if there are relevant metabolite biomarkers in breath for diseases of interest. So this atlas is useful in that latter part, especially the services. And we have that growing database of VOCs known to exist on breath. These are differentiated from background contamination. And we have quantitative data now on those molecules in human populations, and that's only going to grow as we do more studies. So this atlas basically firstly allows confident identification of biologically relevant molecules in untargeted work, but it also helps us to move towards what we want to move to, which is a more targeted methodology where we can focus on VOCs that look like they're relevant in particular diseases and particular biological processes. Of course, having this database also allows data mining across populations and methodologies. So it helps us to understand the underpinning biological pathways involved in generating these VOCs. But it also helps cross-validate results by allowing us to compare to reference populations. And then finally, I think particularly relevant for this audience, we can see some molecule classes that maybe aren't reflected so much in the fairly broad GC-MS platform that we've developed so far. So this is also helping us to try out different GC mass spec methods, different GC columns specifically, to expand that method and be able to probe an even wider range of molecules than we already can. So thanks for your time.
Thank you very much, Elizabeth.